we would like to present the practical on the isolation and identification of Staphylococcus aureus. Staphylococcus aureus is a gram-positive bacterium that appears as foci arranged in grape-like clusters under the microscope. This bacterium is commonly found on human skin and inside the nasal cavity. Although it is normally harmless, S. aureus can become pathogenic and can cause various infections, such as skin infections, food poisoning, pneumonia, and bloodstream infection. Therefore, the identification of Staphylococcus aureus is very important in clinical microbiology to ensure proper diagnosis and suitable treatment for patients. Laboratory identification of this bacterium involves several microbiological techniques, including observation of colony morphology, gram staining, and biochemical tests such as the catalase test and coagulase test. These tests help differentiate S. aureus from other bacteria. In this practical, microbiological methods were performed to isolate and identify Staphylococcus aureus from sample. The objective of this practical is first to determine the count of S. aureus in food sample, second, to practice isolation techniques using selective and differential media, and third, to practice safe procedure for handling pathogens in the laboratory. Moving on to the practical on the detection of Pseudomonas in food processing environments. Food processing environments may contain various microorganisms that can affect food quality and food safety. One important microorganism is Pseudomonas, which is a gram-negative bacterium commonly found in water, soil, equipment surfaces, and food processing areas. Pseudomonas is known as a sponge microorganism because it is able to grow at low temperatures and can cause unpleasant odor, slime formation, and food deterioration. Due to these characteristics, the detection of Pseudomonas is important in monitoring hygiene and sanitation practices in the food industry. In this practical, microbiological methods were used to detect the presence of Pseudomonas from samples collected in the food processing environments. These methods help to evaluate the cleanliness of the environment and the effectiveness of sanitation procedures. The objective of this practical were: first, to practice sampling techniques in the food processing environments. Second, to determine the total plate count, yeast and mold count, and Pseudomonas count in selected food processing environments.
First, the sample site is determined. For group 6, the sample site is at refrigerator door at FSMA ground floor. Then, the swab head is moistened in the solution before gently swipe it on the surface. The swab head is then swiped in 3 directions, which is vertical, horizontal, and diagonal on 100 cm square surface, which is 10 cm by 10 cm. The tube that contains the swab head is labeled as 10 power of 0. Then, we do the first dilution by transferring 1 ml of solution that contains the swab head to other tubes. The tubes is then labeled as 10 power of -1. Then, 1 ml of solution from tube 10 power of -1 is transferred to another tube. This tube is labeled as 10 power of -2. Then, spread 0.1 ml of sample 10 power of -1 and 10 power of -2 onto the plates of PCA plates. The plates then incubated at 35 degrees Celsius for 24 hours. The same action is done for BPA plates. The BPA plates are incubated at 35 degrees Celsius for 24 hours. First, the sample site is determined. For group 6, the sample site is window of female prayer room. Next, the swab head is moistened in the solution before gently swipe it on the surface. The swab head is then swiped in 3 directions, which is vertical, horizontal, and diagonal on 100 cm square sample surface, which is 10 cm by 10 cm. The swab head is then left in the solution after done. The tube that contains the swab head is labeled as 10 power of 0. Then, we do the first dilution by transferring 1 ml of solution that contains the swab head to other tubes. The tubes is then labeled as 10 power of -1. Then, 1 ml of solution from tube 10 power of -1 is transferred to another tube. This tube is labeled as 10 power of -2. After the dilution is done, spread 0.1 ml of sample 10 power of 0, 10 power of -1, and 10 power of -2 onto the plates of PCA plates. The plates is then incubated at 35 degrees Celsius for 24 hours. The same action is done for PIA plates and the RBC plates. The PIA plates are incubated at 35 degrees Celsius for 24 hours. RBC plates are incubated at 30 degrees Celsius for 5 days.
Next, we are going to discuss the results of the isolation of SRS. Firstly, the results of refrigerator surface are 12 counts on negative fine dilution and 1 count on negative 2 dilution. This proves the technique is executed accurately. The negative fine dilution gives results of 1,200 colonies from unit per milliliter, and the negative 2 dilution plate gives 1,000 colonies from unit per milliliter. These values are considered consistent. These counts are low value, which suggests while the refrigerator surface are not sterile, it is also not heavily contaminated. There are several types of bacteria that is possible on this plate, like Bacillus or Listeria, which are psychotrophic, which means they can survive at low temperature. And that makes sense since we swap side is the refrigerator surface. Next, the BPA plates are selective mediums that use inhibitory agents. This means it is a specific medium for isolation of SRS. However, since the results are no growth on both dilutions, this means the one colony on previous PCA plates are most likely not SRS. So there is no SRS growth on the refrigerator surface. Secondly, let's discuss the Pseudomonas isolation. We sample from the window as our swap site. Obviously seen here, it is TNTC too numerous to count on all dilution. This proves the window has high microbial load and heavily contaminated with bacteria. This is probably because the window collects dust and bacteria from the surrounding air. Too numerous to count on PCA plate indicates there are growth of environmental molds, yeast, or hardy gram-positive bacteria like Bacillus. Here are the closer looks for each plate. Too numerous to count indeed. After that, we execute dilutions for PIA plates, which are no growth for all dilutions. This confirms that none of the thousands of bacteria living on that window were Pseudomonas. PIA inhibits almost everything except Pseudomonas. The window likely harbors environmental extremophiles like Bacillus spores. This is because Pseudomonas are hydrophilic bacteria, which means they thrive on moist area, which the window are clearly not, since they show no growth. So, while the window is extremely dirty based on PCA results, it does not harbor Pseudomonas specifically, which can cause food spoilage. Lastly, this is the result for DRBC plate. For zero dilution, there is growth, but no individual distinct circles because the bacteria have grown together, which means it is a confluent growth. This proves the sample has very high concentration of spores. Next, the negative one dilution results in 1,200 colonies from unit per milliliter. This proves there are growth of fungi, but they are not Pseudomonas. After that, there are no growth on negative 2 dilution, which suggests a clean dilution technique. As seen here, the fungi seems fuzzy, which indicates it may be mold spores, and in food science, these spores can cause an atmosphere spoilage for products that are left exposed in that area.
In conclusion, these practicals were successfully carried out to detect and identify Staphylococcus aureus and Pseudomonas using standard microbiological laboratory methods. Throughout the experiment, proper aseptic techniques, sterilization, inoculation, and incubation procedures were very important to avoid contamination and ensure accurate results. From the practicals, SRS was identified based on its morphological and biochemical characteristics, while the detection of Pseudomonas showed the importance of environmental monitoring, especially in food processing areas. The presence of these microorganisms demonstrates why proper hygiene, sanitation, and food safety practices must always be maintained to prevent contamination, food spoilage, and possible health risks. Besides that, these practicals help improve our understanding of microbiological procedures, laboratory handling skills, and microorganism identification techniques commonly used in food and clinical microbiology laboratories. Overall, the experiments emphasize the important role of microorganisms in public health, food quality, and infection controls. They also highlight the need for regular microbiological testing to ensure safe laboratory practices and safe food processing environments. That's all from us. Thank you.